Hello, this is Dr. DeShavo. I am beginning our journey with pathophysiology at the level of the cell. This is Chapter 1, Video 1, and let's get started. General definitions and concepts. This is a general introduction to pathophysiology. This is the preface before Chapter 1 in your book, if you would like to read along with your book as we go through these notes. I want to note once again that the questions for your quizzes and your exams will come directly out of these PowerPoints and out of my lectures. If you look at your book, there will be just about the same amount of information, but I just want to clarify I'll take all exam and quiz questions directly from these notes and from my lectures. General concepts. I hear that collective sigh saying, oh my goodness, why do I have to talk about cellular processes? But I guarantee you that if you gave me the most obscure disease process, I could back it down to the cellular processes that happen that cause that disease. The cell is the basis of all disease processes. So we do need to go over their basic function, structures, and how they interact with each other as well as their environment to understand diseases. Now, pathophysiology, I think you probably understand, is the study of the disorder of the breakdown of human body's functions. This is a little different from what people call pathology. So if you send something to pathology to be examined, that is different from the actual book study of pathophysiology. Don't let that differentiation blow your mind. They're along the same lines, right? But just to sort of give you the right terminology, that we're going to be studying pathophysiology, you'll be sending things to pathology to be examined. Now, I don't know if you remember homeostasis or not. Homeostasis, I always think of as justice, blind justice with the balanced scales. When you're in homeostasis, you're balanced. I have my arms out to the side right now and I'm balancing, okay? You're in balance. When we have a disruption in homeostasis, it can lead to different disease processes. And if we move on to homeostasis, of course, this is dynamic. We are ever changing functional structures. Homeostasis is not stagnant. We are constantly reacting to our environment, to people around us, to our mental responses. So homeostasis is something that will constantly change in order for us to, or have changes occur to it in order for us to keep functioning properly. It blows my mind. If you really think about it, we are such delicate creatures. If we were to change the atmospheric pressure too much, if we were to go to Mars, if we were to be shot off in space, we're pretty delicate. I mean, this is it, guys, living on this earth. And if we were going to get too many extreme environmental changes, our body has difficulty balancing to that. Okay, so we need that equilibrium. Now, the thing about, and I think the rest of this is pretty self-explanatory on that left column, but I do want to talk about equilibrium or homeostasis for cells as well. Once again, the cells are the basis of the whole body. It's very important for cells to have a good, balanced environment in order for them to function properly. And they need to maintain homeostasis, just like we do. Okay, so self-regulating. Let's talk about negative and positive feedback. Okay, every anatomy and physiology book and pathophysiology book brings up negative and positive feedback. I find them slightly confusing, right? Because both of them at some point will stop. Okay, right? I mean, positive feedback, for example, is going to be contractions of labor. That's going to stop eventually, right? So this is what I want you to know. I want you to know an example of both and understand that negative feedback is called such because it will have an opposite response that will balance it. Okay, so for example, you eat some sort of sugar, you're going to release insulin, which will decrease blood levels of glucose or dextrose, and put that sugar into the cells. Okay, so that's a negative feedback system. You have the glucose, you have the insulin puts the sugar into the cells or glucose into the cells, and it balances sugar levels in the bloodstream. Okay, now positive feedbacks, an example is contractions. The difference is you're going to perpetuate those contractions during the event, making it a positive feedback. I find it confusing because eventually labor, thank God, ends, right? Everybody who has had children and are female are like, that's right, thank God, right? So don't blow your mind about that, okay? Negative feedback theoretically causes a negative reaction during the event. Positive causes a positive or a perpetuation during the event, okay? And I will only ever ask you those two examples, the glucose and the contraction of labor, because I do think they're slightly confusing, okay? Now we have several things in place to correct imbalances. Think about everything your body has done involuntarily since you got up. It's maintaining glucose levels. All your hormones are regulating unless you're going through menopause like I am. I'm just joking. I was trying to give you silly things to remember. All of these things are maintaining homeostasis without even thinking about it. Okay, it's a pretty amazing event. So factors that determine baseline normal, I think you guys got all of these. Okay, now how they determine these baselines is they studied thousands of people. They did thousands of blood pressures in order to take the average and figure out baseline normal. They did thousands of levels of cholesterol in order to determine a baseline normal. There are some variations. For example, I know my temperature is about 97 normally. I just run slightly low. It's how I am. So if I'm up to 99, I've got a pretty good temperature already. You know, so there's some little variations of that, but you know, they establish a generalized normal so we know where to start from. And of course, those normals will vary according to, you know, age, gender changes, etc. Okay, geographic area might not be something you think about, but that can determine a different baseline of normal for our patients. Altitude, this is an easy one to give you an example. The higher the altitude, the less oxygen we have. The less oxygen we have, the more red blood cells we're going to make. So their blood's going to be slightly thicker, not a lot, but we'll do that when we go into the study of blood. Okay, and then everything is relevant, of course. If somebody has a slightly high blood pressure, they don't immediately get diagnosed with high blood pressure. We need to maybe see what their normal is. See if they normally get nervous at the doctors. That could be an issue as well. Okay. So pathophysiology is a study itself, just some generalized questions here. Now, etiology is a cause or reason for the event. I don't know if you remember this or if you use this on a daily basis. It may sound very familiar. So what is the etiology for that disease process? Somebody's a vegetarian, they have low B12. That's the etiology. The fact that they're a vegetarian, they're not supplementing with B12 supplements. Now, idiopathic is of an unknown cause. We have a friend who has idiopathic hypertension. He just has it. We don't know why he has it. It's probably genetic. Okay, but just to give you an example of that. Iatrogenic is unfortunately an effect that happens after medical treatment. I'm sure you're all thinking of things that have happened. You've got to hedge your bets. And if you're giving somebody a treatment for one thing, they may have a side effect that trumps or that the treatment trumps, or wins over. Pathogenesis, genesis always means the creation of. So the development, evolution of a disease, the stages that it goes through, and we're going to go through many of those. Some are pretty common sense. Some, like the stages of syphilis, don't seem correlated and seem very divergent. Clinical manifestation, signs and symptoms, you're aware of that. And the difference between acute and chronic. Acute is going to be the initial event short-lived, just for a few weeks, versus chronic when somebody has something long-term. Something can just be acute or can eventually continue and become chronic. Epidemiology is the patterns of diseases studied in a group of people. So if for example, someone says, oh, this group of people has a disease such as malaria, oh, why is that? Well, they live in an area where there's more, mosquitoes that cause that malaria in that pattern or that disease group. So not disease group, that group of people. Okay. So epidemiology is going to study events that happen in groups of people to see why that happens in that area or that region. All right. Now, about every 10 minutes or so, we're going to have a question. See if you're paying attention. No, you're not going to be actually tested on this. There will be weekly quizzes, but I want you to pause for a minute here and think about this question and answer it for me. Okay. And I know we're not in person, but I really want you to think about these videos as if we are actually in class and I'm asking you this question. So in order to understand pathophys, we need a solid working knowledge of which of the following go through. I'm not going to give you the answer until after you actually answer it. Okay. And then we're going to move on to the next after you take a minute and that. Excuse me. Okay. So cellular function, moving on to cellular function itself. Yes, we do need to study these little buggers. Okay. And interestingly enough, cells kind of are like little people. They breathe, they excrete, they create. It's kind of cool. So let's start to go through those cellular attributes. Cellular attributes include the ability to exchange material with their environment. They're able to bring things in and bring things out. They would need to if they're alive, just like we need to bring in oxygen and food, and we need to excrete urine and feces. So cells are going to need those same intake and outtake of materials. Ability to obtain energy from organic nutrients. Whoa, organic nutrients. Okay, glucose and oxygen are key. So let me back that up. So glucose is really going to be one of the main food staples that we talk about at a cellular level. Cells are going to need proteins and fats as well, but let's sort of just go through as they come up what's necessary. And as you know, or maybe you don't know, but let me refresh your memory, glucose is a carbohydrate, and glucose comes from sugared foods. Okay, you can break it down. It doesn't need to be sugar. My kids always say this, but I need energy. No, you can get glucose from fruits, from breads, substances such as that. And what our body does is we ingest that food, we break it down into little particles, and we release glucose into the bloodstream to be absorbed into cells. Okay. Now, the other thing that we need to make energy is oxygen. When we have glucose and oxygen in our bloodstream, we can put both of them into cells, and the cells can actually compile together what's called ATP. Don't worry about it. I'm going to get into it, which is little packets of energy. But glucose and oxygen are super important. This probably makes sense to you. Think about people who don't have enough glucose, who don't have enough oxygen. They can't create energy. Their brain starts to malfunction because they're not creating energy for their brain cells. They start to lose energy. They start to get a little loopy, right? So anyway, just to sort of back up, because that's an important concept. You need glucose, you need oxygen to go into your cells to create energy. Now, our cells also create bigger, more complex molecules. It's how we create the proteins, structures of the body. Okay, and we'll go over a little bit about how cells do that. Cells have to replicate. The process of mitosis is the ability for a cell to replicate to create more cells. For example, our skin cells. You are excreting skin cells all the time. What do you think is the dust in your house, right? So those skin cells have to mitose and create more skin cells in order to replace the ones that have sloughed off. Okay, we've got to replace cells because they get old and they wear out, and we need to create more. Now, the parts of the cells, I'm going to go through and tell you what each part that I want you to know, and I really simplified it. Your book makes it a little more complex than I'm going to make it. I went through and I gave you definitions of the cells, cell parts that I want you to see. And here's a picture of it. So let me see if my drawing, oh, it works. Okay. So we've got the nucleus, which is right in the center. And I think you probably remember that's like the brain. Okay. And then mitochondria. Okay, mitochondrion is singular, are those kidney bean-shaped cells that create energy. I don't know if you remember that from A&P way back when. Okay, and then we have endoplasmic reticulum. We're going to talk about rough endoplasmic reticulum. And I always think of that as being these folds. It always looks folded, like folded membranes. Okay, and on there you can see, if you look a little closer, there's dots, and those dots are what create proteins. This is all in the notes, okay? So don't be panicking that I'm going very fast. I'm just giving you visuals right now, okay? Lysosomes actually have substances in them. They're little round cells that help to break down substances in the cells. We'll talk about those in a little bit. There's different microfilaments. I really kept that simple in the notes. I'll tell you what we're going to talk about, one of which is cilia. And everybody remembers cilia. Cilia are those finger-like productions on the surface of cells. They help to move substances along, but they also help to filter. We'll talk about in the respiratory system. Ribosomes are the dots that are on endoplasmic reticulum, which help to create proteins. Flagellum is only found in sperm. It helps to move the sperm forward. If you have not seen videos of just the sperm, nothing naughty, just the sperm moving towards the egg and reproduction, it's fascinating. These flagella, which is plural, flagellum is singular, move the sperm forward very quickly and in a strong motion towards the egg. It's just unbelievable. Okay. All right. So that is basic structures of the cell. We're going to go over the functions of them in the next few slides. And I recommend, as we're going over those functions, go back and just get a visual so you can remember what they look like. Okay, 3 major components of eukaryotic cells. There are tons of components, okay? And we talk about eukaryotics are actually ones that have a nucleus as opposed to prokaryotic. Don't worry about that. You may remember that from general bio that don't have a nucleus, but eukaryotic cells are going to have a nucleus, which is the control center. Now, guys, this is the control center that has all of your DNA. DNA is what creates us, is what makes us all individual. It holds and encodes our genes, which tells our eyes to be brown or our hair to be blonde or the height of our body, okay? Now, the other thing that's important with DNA, make note, is it contains the codes for all the proteins of the body. Okay, so not only does it hold the genes, it holds the codes for all the proteins of the body. So if you need to make more hair, okay, the genes in that area that create the hair create the strand of proteins that will create that hair, okay? Any of the structure of the body we're going to get from the DNA that is in the nucleus. So not only does it create the genes that tells our body what it's going to look like and how we're going to act, it also holds all the codes for all the structural proteins of the body. So make note of that, okay? The cytoplasm is just all the water, okay? There's also some protein and fats in there. There's glycogen. Make note of this. Glycogen is a stored form of glucose. So in the cytoplasm, we will have long strings of glucose units connected together. That's called glycogen, okay? It's just the way we store it for energy. And in the cytoplasm, we also have organelles, which are teeny tiny organs, okay? Basically function as organs. And they're all those structures that I went over briefly before. Mitochondria, lysosomes, endoplasmic reticulum, etc. So you have the outside of the cell, you have the cytoplasm with all the organelles, and then on the inside you have the nucleus. And once again, if this is starting to seem a little fuzzy, I want you to pause it and go back to that picture and look at it again. Now, the cell membrane is semi-permeable, which means it has what's called a phospholipid bilayer. I don't know if you remember that from A&P or not, but only fats, or very small things such as oxygen can pass through the cell membrane, but that's a good thing. We don't want everything coming in and out, okay? What it does have on the surface of it are little protein receptors. And so we have, if we move down, I'm going to jump down here a little bit. Let me see if I have a picture of it. Sorry. Okay. I don't, but I'm going to get you one. Okay. So I'm going to get you a better picture of the cell membrane. I'm going to put it in a separate resource area for you. So we have the surface of the cell itself, which if we do move down to the next picture, is a phospholipid bilayer. Okay. And here's this phospholipid bilayer. And the center portion that I drew the arrow to is made up of fats, and the outer portion is a phosphorus. You don't even need to remember a ton of that. But what that does is it makes that center portion that I just pointed to impermeable to anything except fats, and usually small fats. And this outer portion really likes to interact with the watery environment. So that way it doesn't reject the water that's outside and also inside of the cell. Okay, so this would be the outside. This would be the inside, okay? Now, semi-permeable means that only certain things are allowed in and out. Think about it. If we let everything into the cell, it would swell up and burst. If we let everything out, it would shrink. We need to keep it a nice, healthy size in order for it to function properly, okay? So small substances can pass through. I think oxygen is a good one to remember. You don't need to remember all these ones listed here. Okay, hydrocarbon, you don't even need to remember that. But you can remember fat or lipid soluble. Lipid is another word for fat substances can pass through. Okay. Large water soluble substances, as we see in this third slide here, cannot pass through. Okay. Ions cannot pass through as well. Okay. Sorry, hold on. It likes to do this occasionally. Okay. So let me stop this. All right, there we go. All right. So only certain things can get through. Now, what we do have that passes right through this membrane, it's almost like a channel, are protein receptors and channels. And they pass right through, and they're a solid protein structure, basically with a hole in the middle. So suppose we want to get glucose in, it can pass through very specific glucose receptors into the cell. Suppose we want to get sodium in. There'll be another protein receptor that passes right through the membrane that sodium can pass through. And that also allows very specific substances to pass through at very specific times if we need them. Okay. Functional cell components. Ribosomes are the site for protein synthesis. So when we want to create structural proteins, either for the cell itself or to send it out to be used in the body, such as muscle or hair or skin, the ribosomes, which are those little dots on the endoplasmic reticulum, are where we do it. Okay, riboplasmic reticulum can create other substances like fat and enzymes, but it's primarily used to store those ribosomes, and it creates a structure for them to be on where we create those proteins for the body. Golgi apparatus helps to package substances such as those proteins to be excreted from the cell. I always used to call it the UPS of the cell to, package and send stuff out. Lysosomes help to break down cell products and also foreign bodies. If we were to get some sort of virus or bacteria, lysosomes will release enzymes that will help to break down either used cell products or those foreign bodies that we can either be used again or break them down to be excreted. Okay, peroxisomes help to break down toxins in the cells. Okay, it actually has almost like a peroxide-like structure or substance in it to break down toxins that we have in the cells, to protect the cells. Okay, functional cell components, mitochondria are the power plants. That's where glucose and oxygen combine to form ATP. We talked about cilia and flagella. They're hair-like processes that help in movement. Flagella particularly for movement. Cilia can help with movement, but also with filtration. We have cilia in our respiratory tract that help to keep substances out of the respiratory tract. Cell component membrane receptors, okay? And I'm going to get you a picture of protein receptors on the membrane. Those are channels that go across that membrane that I drew on the picture above that allow certain substances to come in and out. They can also send second messengers, which we'll talk about when we get into hormones, okay? All right, another question time. And of course, if you have any questions, shoot them off to me. And your question right now is the cell's semi-permeable membrane allows it to maintain a proper shape. Is that true or false? Okay. And at this point, I'm going to pause the tape. I'll start creating the next tape and you can fire it up and get going. Please, as always, if you have any questions, let me know. Take care.